Well, good afternoon, everybody who decided to join Sano Seminar on computational medicine. Now is 5 minutes past 2, so it's high time to start our seminar. And it is a seminar from the series of Nobel Prize seminars at Sano. And fortunately for this Nobel Prize, we have a very good speaker. The speaker is Tomasz Kościółek, who is a leader of a research team at Sano. And he's leader since practically 1 year and a half. Before that, Tomasz was graduated in chemistry from Jagiellonian University, and then he has got a PhD in biology from University College London. And he spent several years at different places in States and in Europe. He was also a group leader and bioinformatics at the Małopolska Center of Biochemistry. And he also collaborated with institutions in Poland. Well, and today he will explain for what this Nobel Prizes chemistry were given. Tomasz, screen is yours.
Great. Thank you, Marian, for your very kind introduction. Hey, welcome, everyone. Thanks for everyone who's in the room. Hello. Thanks to everyone who's online. Folks, let me level with you. Nobel lectures, so lectures given by the Nobel laureates are out. Here's a link to a YouTube video. It's also 1.5 hours. So if you'd rather get it straight from the horse's mouth, just go and watch it. I highly recommend. All 3 Nobel Prize laureates in chemistry are really good speakers. And I'm sure I haven't still watched all of it, but I'm sure those lectures are great and they will be very informative. What I'm focusing on today instead is my take on why I think this breakthrough was possible. And I will tell you this through the lens of 3 stories of 3 different people, our laureates. So let's go back 10 years, almost to the day, December 2014. Demis Hassabis, the CEO of DeepMind, was giving a lecture to a group of UCL students. He was telling the students about the recent acquisition that happened at that time when DeepMind was acquired by Google. At that time, still Google was a very revered company, maybe not controversial. And at that time also, there was a lot of mystery surrounding that acquisition. People were very curious to learn more about this. So Demis began his story by describing how, as a child, he really got into chess. In fact, he was so good, he was a child prodigy, that he became a chess grandmaster. But as he got older, he became interested in other games. He became a game developer as a teenager, where he joined Bullfrog, a game studio. He's one of the main people behind games such as Theme Park or Syndicate. So for those of you who don't know, Theme Park was maybe one of the early versions of Sims or other type of like simulation games that one could play to build an environment and then see how stable it is and how it develops. Then clearly Demis is a very bright kid, right? But he's also a nerd. He really likes games. He's very much interested in how things work and how brains work and what we can learn from that. So much so that he got a PhD and he became a postdoc at the Gatsby Institute, UCL. It didn't take him long until him and his colleagues from Gatsby established a company, DeepMind. It was himself, Mustafa Suleyman, and Shane Lay. But also it didn't take long for DeepMind to get acquired by Google, because it was just 4 or 5 years until that really happened. And their objective, the objective of DeepMind, was really simple. Develop artificial general intelligence as something that we hear of today, maybe much more, but it was way more shocking back in 2014. But the way that DeepMind wanted to go about it was maybe a little bit unique. So Demis, throughout his life already, was very much well-versed in different types of computer games. So much so that what DeepMind started with was trying to find solutions to simple games. It's Pong. Initially, the computer doesn't know anything. It's clueless. It doesn't know how to move up, how to move down, but what it knows is the score. So by learning, the computer in time can figure out how to play Pong better and eventually master it. That was one of the first challenges that DeepMind embarked on. Later on, well, unsurprisingly, they moved on to other types of games. What is so special about games except that they're just simply fun? Well, with games, you can repeat the learning process essentially up until infinity. You got a set of rules that work within the world of the game. And well, you can measure usually either in a very simple way, using the example of Pong, or in a more difficult way, you can measure the progress that you're making. So through this example of different games and perfecting the methodology on games, DeepMind was having a go at another problem. In time, they moved to complicated games, just Go, StarCraft, so on. At that time, in 2014, the progress was that DeepMind is really good at teaching computers how to play and be really good at different games. What DeepMind was really about, and what they were looking for, is then how to address a problem that is actually meaningful and impactful. And one of those problems, and the definition of the problems that Demis himself gave, was that it needs to have a massive combinatorial search space. Just as it's any game, you need a clear objective function that you can optimize for. And you need enough data or a way to simulate your game environment so that you can learn. And this 2014 date was not maybe that random, because Demis and the acquisition by Google and then the subsequent move that happened to other quite important spaces. So one of the challenges that DeepMind soon thereafter realized was that there exists such a problem in biology. And this is a problem of protein structure prediction. So we do have a what are proteins and their shapes, and why do we even want to predict them? So the problem in principle is quite simply defined, right? So a protein is a chain of amino acids, and there exists a very finite amount of 20 different amino acids. And as we write the sequence down, it corresponds to a unique three-dimensional structure. And this three-dimensional structure of a protein is really important because this is how the protein enacts its function. So for those of you who are maybe not that well versed in biology, proteins are one of the main types of molecules that are actually responsible for enacting biological function, for molecular transport, for processing of energy, and so on and so forth. So unlocking and really understanding this language of proteins is something that is very prescient to understanding biology and to solving biological problems. But not only that, that is a problem that is also well defined. But before I go to that, let me just show you a little bit what different types of proteins may look like. So they come in all shapes, forms, and sizes. So we've got the squiggly bits that are helices, and this is one of the types of secondary structure. So this is the main form of organization that the protein can take. We also have loops that don't have any particular local order to them. And then we also have beta sheets, which are usually represented as in the form of arrows, and those are usually perpendicular. For example, as you can see here, perpendicular flat parts of protein. But also, as you can realize from looking at this picture, the diversity and the size of proteins is immense. And this is really speaks to the scale and importance of this problem. And the big problem it was. So in green, I'm showing you the number of known protein sequences. So those are those strings of letters that we can infer using different experimental techniques that today are mostly high throughput. So we're getting a lot of information, we're accumulating a lot of information very quickly. And this is reflected by the size of the UniProt database. So back in 2020, it was roughly 180 million of different sequences that we had access to. But what happens if we look at structures that were determined experimentally? Well, and using the same scale, the line is essentially flat and it didn't change ever. But we zoom in, we actually see that there is also a very stable progress in accumulating different protein structures. But back in 2020, that was 160,000. So as compared to the number of known sequences, this number was a really minuscule amount. So in terms of our understanding of protein structure and our understanding of biology, this was still very limited. And mind you, those structures are obtained using experimental techniques, be it X-ray crystallography, NMR, or nowadays more and more using cryo-electron microscopy. But the challenge here is that unlike the sequence characterization, the obtaining the strings of letters that is cheap and easy, obtaining each protein structure is and was quite challenging and expensive. To get and characterize protein, you need to obtain its structure, its crystal. Well, first it needs to be clean, then you need to obtain its crystal, and then only you can go to synchrotron or another diffractometer and obtain the X-ray diffractography. So essentially, each X-ray structure was, well, depending on the scale of the challenge, but we're looking at kind of one structure per PhD type of challenge. So this was very, very laborious. But also, at the same time, What happened in the community working on bioinformatics, so trying to understand biology by the use of computers, was a deep desire to really address this problem, because it was proven by Christian Anfinsen originally in the late 1960s, for which he got a Nobel Prize in chemistry in 1972, was that the sequence of a protein corresponds to its thermodynamically optimal. The protein sequence falls into a well-defined thermodynamically optimal three-dimensional structure. So a protein three-dimensional structure is determined from its sequence. And not only that, it would correspond to its free energy minimum, which is very important information. To that end, a really important thing happened in the bioinformatics community in 1994. So it was John Molt's initiative. John Molt from the University of Maryland himself, and who was the director of this working group, was Krzysztof Fidelis from UC Davis. It was a critical assessment of techniques for protein structure prediction. This sounds quite obvious today, but that was the first of its kind type of community-driven evaluation that was happening every 2 years to study how computational methods for protein three-dimensional structure prediction, how good are they? Are we getting any better over time? What are the challenges and what are the solved questions? And the way that CASP worked and still works to this day is the organizers of the competition. So this is an independent evaluation, right? Every single group that decides to participate in CASP does not know the answer until after the competition is completed. So the organizers of CASP contact experimental groups and ask them, hey, what are you working on? Or what is the structure that you're expecting to determine in the next few months? And as they go through different groups, then they aggregate all those different sequences. Then they try to split those sequences into different groups, what they expect to be difficult. This is a very long protein. It's obviously going to be way more difficult to computationally predict than just a small one. Is this similar to something that's already been known and is already in the databases? Or is this something that's potentially just entirely new? And under those conditions, participating groups create models of protein three-dimensional structures, and then this is all being fairly evaluated by the community. How did it work? Well, in 1994, it was terrible. 1994 and CASP1 essentially predicting a protein structure for something that is unknown was impossible. There were some reasonable guesses as to what is similar to some known protein or not, but really what protein structure prediction works for at the time was very easy targets that could be identified by just similarity to something that was already there. Well, that's a start. Everyone needs to start somewhere, right? But over the years, and this is a more granular view of the changes, so it's easy to see. So that was CASP1. Nobody basically had any idea what to do with this protein. But CASP2, there was progress. And then over the years, we see gradual improvement, but there are a few quite significant leaps. And I think those leaps are also very significant. Not only CASP alone, but CASP was this community that set up a set of good working practices, was a community that helped to establish how to measure how well we are performing in predicting protein three-dimensional structures, but also there was a community and a forum to exchange ideas and exchange those best practices. If CASP wasn't happening every 2 years, getting that progress would not be possible. And also, it would not be possible if people did not share the methods and the findings and the ways and means by which they were able to achieve this improved performance. I'm not old enough to know how it might have looked, what it might have looked like in 1994 and 1996 to exchange those algorithms and ideas in the very practical terms, but it happened nevertheless. Okay, so let's move on to the second hero of our story. And here I'm gonna be a little bit subversive because I'm not gonna tell you about one David, I'm gonna tell you about two Davids. I'm gonna tell you a little bit about David Baker on the right here. Accurately, more or less dated photo, so that's David Baker around early 2000s. He's a professor at the University of Washington in Seattle. And here's Professor David Jones from UCL. And an important thing that happened in CASP2 was David Jones's innovation in trying to predict the structure using multiple sequences and recognize super-secondary structural motifs. Sounds difficult, I know, but bear with me. Essentially, the achievement was that for the first time ever, a de novo case of a protein was predicted. This is a very small protein, NK lysine, less than 100 residues. So for some context, we're usually dealing with proteins of a size between 200 and thousands of residues. So proteins smaller than 100 residues are outliers, even there are some peptides, so another biological entities very similar to protein that are in this range. So I think it's fair to say it was one of the easier cases to be solved. Nevertheless, it was a breakthrough because for the first time, it wasn't a method to try in a smart way find an already solved structure, but rather to simulate the structure efficiently enough so that a new protein geometry can be discovered. David Jones did two things here, really. So those are the models here, right? And this is the actual structure. It's not great. Here is a reported RMSD, so root mean square deviation. So it's around 6. Today it wouldn't fly. This is somewhat resembling what should be predicted. But important thing was the use of different types of energy terms. So to try and split and better understand what different energetic contributions sum up to the free energy of a protein following Anfinson's insight. But also it was an attempt to deal with just the sheer combinatorial complexity of predicting a protein three-dimensional structure. Because following Lewinthal's paradox, if the protein folding process is just random sampling, the number of possibilities just very quickly explodes, and it just will be larger than the time since the beginning of the universe, or a phenomenon of that order. But so it cannot be random sampling. We need the computer, the algorithm needs a way to be able to efficiently sift through different types of structures to try and find the optimal. So how did they do it? Well, let's go back to David Baker, because following this initial breakthrough by David Jones, Baker Lab quickly adapted this type of solution and embedded it within an algorithm called Rosetta. And the way that Rosetta worked, similarly to David Jones's fractal, was the using of a fragment library and Monte Carlo fragment assembly, Monte Carlo sampling. So the observation here is we're not treating every residue. So we're not treating every single part of a protein individually. Instead, well, proteins kind of fold into those nice secondary structural elements and beta sheets and all those different loops. It turns out that if you look at enough of them, it's not random, right? We can actually find some small fragments that explain all the different shapes of protein. So instead of using an individual residue as a unit, what they're using is a fragment, so a set of several residues. Now we can achieve efficient sampling in this physics-based approach using a Monte Carlo fragment assembly. So we're starting from an extended sequence, we insert some type of a fragment, and then we check whether this decreases energy. And once we do this enough hundreds of thousands or millions of times, we will hopefully arrive at a structure. But then we need to take account for an entropic effect, right? We're talking about free energy. So this type of simulation from a sequence using fragments, Monte Carlo fragment assembly, using an inverse Boltzmann derived energy function helps us to arrive at another random model. And once we generate enough of those sample models, we accounted for the entropic effect contributing to the free energy function. And then looking at this distribution of different models, we should be able to actually predict the final structure. And again, this is going back to CASP3, so this paper is 1999. So still fairly early days. You can see that those are relatively small proteins. And what you can actually see here is that the native, so the actual experimental structure is shown here on the left, and there you have those indications, model 4, model 5, and so on. So what you could do in CASP initially is submit several guesses as to what you think is the final answer. And what turned out to be a really big and significant problem in those days and for many years to come was actually making the right decision as to how to pick the actually correct model. So actually going back to this plot, one of the major challenges in making this progress from here to here and further was really an improvement. Well, of course, more data becoming available, more computational power becoming accessible. Those all very important contributing factors, but also figuring out better ways by which, without knowing the actual answer, how we can select the optimal model. This also improved over the years. So yeah, here's another take on that. So this is just using fractal, and this is David Jones's program. It's very, very, very similar principle, maybe shown in a slightly different way. And essentially, this type of approach, it improved with better, more defined energy functions, better parameterization as more data became available, better computational solutions. So there was some progress, but indeed the next breakthrough, dare I say, happened once contact predictions were introduced. So what are contacts? Well, contact is, or any two residues within a protein chain that are close to each other. And if we could obtain such a contact map by not doing any simulation or prediction, well, that's wonderful, because actually this contact map already contains a lot of the information about the geometry of the protein. What happened was really the development of a few key algorithms that looked at sequence. Those algorithms look at sequences and looked for patterns of correlated mutations to try and arrive at a contact map that would constrain this search space. So the idea and the thinking behind this was as such. So our access to sequence information is much easier than access to three-dimensional structural information. So getting this is cheap. And what we're getting out of a multiple sequence alignment is really evolutionary information, right? By aligning multiple sequences of related proteins, we're backtracking the evolutionary history of this particular protein. And the hypothesis is that, well, if there are patterns of correlated mutations, which means that, well, if a change happened in a single place, then a compensatory change will happen in another place that was in contact to actually maintain it. This initial observation seemed to be correct, or at least it seemed to be efficient. So using contact information as an additional constraint on the search space was a really powerful way by which those already existing algorithms could be improved. And in CASP11, it resulted in a few kind of very good results and a few contributions from, well, the usual suspects that gave a sense of hope that perhaps this type of approach, so we're constraining the search space by the use of contacts, and then we have reasonable kind of physics-based algorithms to take care of the rest and predict the structure, three-dimensional structure of a protein. So CASP11 was year 2016, 14. And that's more or less what the situation like, or the state of the art looked like at that time. So it was a collection or a superposition of all of the lessons learned for the prior 20 years, right? Quite complicated pipelines that each of them tried to solve a little part of this grand problem. And here I'm very purposely showing one of the more kind of extreme examples of going about solving the problem. So there was quite a complicated decision tree on how difficult the target is, and then what methods should be used depending on the predicted difficulty of the target. And then there were multiple rounds of simulation, and then clustering, and then resimulation and optimization, and trying to get the physics right, and so on, and so on, and so on. But by that time, we've got CASP11 around this maroon line. Things were improving, but they didn't change very substantially. Until that moment, this black line. What happened here? Well, because guys from DeepMind, they actually became interested in the protein structure prediction problem. So Demis had his list of what constitutes a good problem to be solved by machines, by machine learning, by deep mind. And I hope I already convinced you that protein structure prediction has a lot of those features, right? It's an important problem. It's well defined. We know how to measure. There is a community or environment that supports this, and we have ways and means to check how we improve year on year. And that's exactly what we did. So AlphaFold 1, and let me underline this one, AlphaFold 1, the one that nobody's talking about today anymore, was really perfecting the formula that was developed over decades, but with the best and brightest deep mind engineers. So what did they do? Well, AlphaFold 1 is essentially a variation of what I just showed you before. So they used a deep neural network to use this evolutionary information coming out of multiple sequence alignment, but not to identify contacts, but distances. So this is richer information. And also, they were able to define or predict torsion angles. So what will be the tilt of any given residue within a protein. And this is a very complete picture of a protein, albeit in a slightly different representation. So then what AlphaFold did was a very simple gradient descent to try and now satisfy all of those constraints and come up with a three-dimensional structure of a protein. So that's 2018. And that was the perfection. Formula has been perfected, taking decades of gradual improvements stemming from the community-wide effort of CASP, putting the best engineering minds, putting millions of dollars of funding and all of the benefits coming from improved computational power to bring AlphaFold 1. And this is more or less what it looks like. Was this a big deal? Well, this was a very big deal at the time. So the Times, the British one, Science magazine, and many other scientific and research outlets were writing about this. Why was this so important? Well, first of all, the results that DeepMind achieved at the time were really impressive, right? As you could see before on that plot. But second of all, that was one of those rare instances where actually a crossover happened, right? So a company that 100% stems from academia, but then they made a crossover, became a startup, became acquired by Google, and then they decided to participate in a scientific experiment. That was quite novel, and that was the first occurrence within CASP also that happened. So really a lot more attention came to the problem, and a lot more confusion within the community also came to the problem, to this area. So the question really that started making rounds back then was, well, how can research groups really compete if a company with essentially limitless resources is attempting to address the problem that they've been working on for decades using academic resources? Well, that's not all. Yeah, here's another example that really the CASP-13 results created quite a significant noticeable improvement in structure prediction, but also they demonstrated fairly clearly, right? Because here you can see that, the first and second place were very close together, but in CAPS 13, Google DeepMind, they really showed with AlphaFold 1 that they can master the problem. Now, introducing John Jumper. John got his PhD from the University of Chicago back in 2017. Working with Tobin Sosnick, working on AI-inspired methods to improve molecular dynamics simulation. So what he was interested in was mostly how to use this new fad of AI to try and better simulate, so understand the dynamics of proteins. So for example, to address problems of how ion channels work. So how an ion can travel through a protein, a channel within a cell made out of a protein. And those sets of problems are very important because the dynamic problems, especially for molecular machines, for a lot of molecular complexes, is where really the biology happens. So understanding dynamics was also something very important. But just like previous heroes of our story, John was not a dummy. He did 6 months of postdoc at the University of Chicago, finishing his project. And in 2018, so right at the time when AlphaFold 1 was released, he joined DeepMind as a research scientist. And they got to work. I was telling you over this story so far, I was telling you a lot about the value of community experiments and the value of gradual improvements. How from CASP 1, this misery of not being able to predict anything bred this innovation of looking at doing fragment-based assembly, inverse Boltzmann energy estimations, and then year over year, CASP over CASP, gradual improvements. When John arrived at DeepMind, they started from scratch. They started from looking at phenomena and not solutions that were already there to address those phenomena. And I think that this is a good illustration of the core of what they really serve. So what they wanted to create originally, moving from kind of building on top of AlphaFold 1, was to understand the language of proteins, right? So the sequence determines the protein three-dimensional structure. So there exists kind of a language of proteins, right? There must be. The first take was to look at language models. That didn't work very well. But what is central to addressing and understanding the protein structure problem? Well, a significant portion of information comes from evolution. It's been already shown and demonstrated, and DeepMind themselves proved it very well that MSAs, so those collections of sequences, they trace evolution really well, and then this can be leveraged to better understand the distances between residues. The second thing that they started essentially from scratch addressing is question of, well, do we need physics? And they figured that, well, maybe those kind of inverse Boltzmann energy terms that are, essentially statistical potentials, right? They're derived from statistics. Well, maybe we don't need to really be defining it by hand. Maybe the algorithm can figure out physics by itself. That's what they tried. AlphaFold 2 and this is what you're looking at right now, was nearly, in my mind, was nearly total rethinking of how to approach the problem. So instead of just adding on top of what was already there, they took a fresh look. Right. So of course they take a sequence, or there is a database of other sequences that exist in the world. From that, you can construct this multiple sequence alignment. And this gives you this evolutionary information. But then we had all those decision trees and whether this is difficult or whether it's an easy target, whether we've ever seen something similar to what we want to predict, or whether this is a completely new, potentially entirely new protein structure. Well, how can we address that? Well, you don't need a decision tree to take advantage of that. Let's just search the database for everything and anything that's similar to our initial sequence of interest, and just represent this as a let's have a pair representation, and then let the algorithm kind of figure on its own how useful that is, or whether this can be utilized efficiently. And then we're coming into this Evo former box that exchanges this information. Does the evolutionary derived information agree with what we're seeing from the initial guesstimate of the structure? Yes, no, let's exchange information. Let's exchange information. Let's exchange information. And then once this information is being exchanged, there comes a structure module that tries to figure out the physics for what you want to predict. And lo and behold, at the end of this process that requires just a little bit of search and then essentially a deep learning part, we're getting from an input sequence a three-dimensional structure. Here are the details of the Evoformer, which probably I will not go into. Again, I recommend the Nobel lecture. And here is the structure module that, I don't know, maybe to folks who are more kind of deep learning first these days, this is more obvious. But that part was really shocking to me. That starting from just a backbone, kind of a basic orientation, and the residue gas. So just atoms floating in space corresponding to what particular amino acids should look like as a chemical molecule. A fold going through this structural module is actually able to figure out physics, right? It's never told that a 6-member ring looks like this and this is flat and so on. But oddly enough, or not surprisingly enough at all, it gets all those things right. How right? Well, very right. Because this is the change that happened from AlphaFold 1 to AlphaFold 2. And this is why everyone keeps saying that, including the Nobel Committee, that the protein structure prediction problem is solved. So what are we seeing here, actually? Well, first of all, this is the GDTTS. So this is a measure. This is one of the achievements of the CASP community, finding one of the good ways to represent and measure how well we are doing in comparison to other methods. And essentially, a GDTTS of 80 or above means that the structure that you're getting is difficult to distinguish from the experimental one. So in essence, what happened, say for 3 examples with AlphaFold 2 during the CASP experiment, was that it was flawless. It was essentially essentially didn't produce single really bad prediction, and most of the predictions that it produced at the time were of really experimental quality. So much so, I remember watching the initial announcement of CASP results, and then watching the keynotes present. And there were 3 keynotes were from experimental groups. And one of the presenters, one of the speakers, said that they've been working on the structure of a protein, and they submitted it to CASP because they thought this would be an interesting thing to do. But soon after, they realized that they're having a little bit of a problems with some experimental work, and they may not be able to actually produce an experimental structural validation on time. But lo and behold, actually the leaders of CASP experiment gave to this experimental group structures predicted or generated by AlphaFold. And the presenter said that the structure created by AlphaFold helped her to actually fix the problem in their experimental setup and their analysis of data to generate the actual final structure, which would otherwise be impossible because they made a mistake. AlphaFold was right. So that's like within 20 years, starting with end of 1990s, where bioinformatic algorithms were able to sometimes predict a small protein up to 100 residues that if you squint your eyes, you may be able to work out something useful from it. All of a sudden, it changed into a situation where actually experimentalists, so usually the people who would just submit, the data for the benefit of bioinformaticians having fun, were really benefiting from this technology. And here, what I'm showing at the bottom is just some example results from the CASP at that time. So you can see that, the GDT score is above 90, and essentially both colors, so the experimental result and the computational prediction, essentially trace each other perfect, with a few minor differences. One of those differences, I don't remember which one, was this key to the experimental insight. And also here at the top is essentially an extraction, not tracing of the folding process, but rather intermediate results showing how AlphaFold is figuring out the structure as it goes. And one more thing about AlphaFold itself and why I think it became so successful that it did, and it ended up in the Nobel Prize as it did. So as I mentioned, one of the problems that was very prevalent in the field for decades was trying to figure out which model is best. Because amongst several alternatives, trying to pick out the best model, if it is a computational exercise, well, this is something that can be maybe evaluated. But then, if this is something we're doing the computation for the purpose of actually using the model to then inform experiments, to draw conclusions from, or to just learn new biology, well, we need to know a priori, without getting actually the experimental answer, how good is the model. And the DeepMind team, they were able also not only to develop AlphaFold 2 by rethinking the entire problem. But also they were able to generate an additional network or to train the entire model so that it knows how good it is. And I cannot emphasize enough how important that is. Because let's take this example right up here. All right, there are parts that are colored and you've got different colors. From blue means good, and so this means that the model thinks that, well, those parts that it predicted are high quality. Then there are some parts that are not necessarily good quality. This can inform experiments. This can inform additional studies. This can inform how this entire prediction and this entire model be treated. Yeah, it informs a lot. It didn't take long for AlphaFold to find its followers. And as you may recall, when I mentioned David Baker's name for the first time, I mentioned Rosetta, and that was the software suite this time for modeling and analysis of proteins. But Baker Lab very quickly, after watching the keynote, they must have been halfway through, because within 6 months they coded up, released, and published a paper with Rosetta Fold that achieved results comparable with AlphaFold 2. Why is this important? Because however DeepMind may be the forefront of even not only deep learning or AI innovation, but also a lot of academic innovation in this space. But they were not always acting like that. When AlphaFold 2 became public knowledge, DeepMind wasn't that keen on releasing it to the public. That did not happen until a lot of negotiations and the pressure from the community, but also because of people who actually started developing very similar algorithm that could have undermined the original DeepMind 10. Rosetta Fold is also particularly important with relation to protein design. This is the other part of the Nobel Prize in Chemistry from this year. I'm going to be talking much about protein design because, frankly, I'm not an expert in protein design. I never did that except for listening to a few of David Baker's talks. But essentially, the crux of the problem is the exact inverse, right? So essentially, protein design is the inverse protein folding problem. So given a three-dimensional structure, what sequence it needs to have, and then by extension, how we should encode it in the DNA to get this protein. So by solving the protein structure prediction, this folding, I think this is an overstatement, protein structure prediction problem, we have an easy way to now also solve or have a much better grasp at the protein design problem. So the Baker Lab, and mind you, David Baker is and thinks of himself as a biochemist, not a bioinformatician. He's mostly interested in solving biological problems. So what they were doing for years and years now, and this is not random, like he's the director of the Institute for Protein Design, at the University of Washington. So what they've been doing is over the years and the decades, they've been perfecting all the different aspects of protein design. And this is a very important also lesson as to how this type of cutting edge research may come about. Because Baker's lab is notoriously very large, like 100 people, not like. Give or take 10 or 15 people who are working on computational problems and computational infrastructure, and then 70 people each working on a different biological problem. So the interaction between the experiment in the wet lab and experiment, the computational experiment, always happens. And this is exactly what is key to the protein design, to addressing the protein design problem. There are some technical details that I don't know to tell you about, so I'm going to tell you about what I know about protein design. So by solving the folding problem, now they were very well positioned to be addressing the design problem. But the important thing here is this iterative process, right? So design has many quirks, right? So nature and real proteins optimize for the process, and they are an effect of millions of years of evolution. So now if we want to create something that does not exist in nature, we need to kind of jump over all the hoops that the nature prepares for us. So the kind of the canonical design process starts with a desired structure, then it goes back to sequence, and then using a known structure prediction method, you do predict a structure from a sequence again, thus making sure that actually this is a viable design sequence. Then the sequence is then encoded as a gene, and in the lab, it's being produced and initially tested for its desired properties. So why on earth would anyone design protein? Well, first of all, because initially the design challenge started because it was interest. One of the first designs was an attempt to create something, a shape of a protein that does not exist in nature or is not described at that time to be existing in nature. And that was indeed possible. But as the challenges got addressed, subsequently, what became more interesting was really the applications that protein design can unlock. That is, we can build artificial protein cages in which we can close different small molecules, different drugs, so that they are being delivered to a specific place in an organism or in a cell, thus requiring lower doses of any given drug to be administered. We can think of protein design solving the problem of enacting different functions for biotechnological purposes or for production of different drugs or enzymes that will have desired properties. Finally, protein design can also be useful in the development of biologics, so protein-based medication itself and protein-based vaccines. What indeed did happen coming out of David Baker's lab in the last 3 to 4 years. So with that, I'm going to be starting to wrap up slowly and look back to the 3 phases of the same problem. 3 phases of the same problem were, of course, David Baker, Demis Hassabis, and John Jumper, the 3 Nobel laureates. Formally, Demis and John got their Nobel Prize for protein structure prediction, whereas David Baker got it for protein design. But I think it's also fair to say that David Baker's contribution to solving the protein structure prediction problem are very significant. Indeed, since protein design is the inverse protein structure prediction problem, it is face of the same problem. Then below, we've got three faces of the same problem. Yet again, the face of Charles Darwin, representing the evolutionary perspective, and that we are really learning from evolution to be able to understand biological problems. The face of Ludwig Boltzmann was instrumental in trying inverse Boltzmann equation. Essentially, was very instrumental to development of the early potentials to Rosetta and Frackfold and other methods. The third phase is the phase of AI that finally brought it all together to a solution. But also there are the three phases of the same problem, in that the protein sequence determines uniquely the three-dimensional structure of a protein, but then the structure of the protein uniquely determines sequence and structure uniquely determines its function. So really, through all this wealth of data that we aggregate through new sequences that are being obtained from a variety of experiments, we have the knowledge of understanding protein structure. We just unlocked it. We awarded the right people. Or did we? Next frontier, well, let's use those proteins for good. With that in mind, I thank you for your attention. Hope it wasn't too boring. To welcome any questions. Thank you very much.
Tomasz, many thanks for your talk. It was a kind of a lecture on this topic, not only about this Nobel Prize in chemistry. Well, we have we already have people who would like to ask questions and take part in discussion. Olaf, well, go on.
Hello. First, Tomek, many thanks for this very interesting talk. And in my spare time, I did actually participate in several CASP rounds. I think one of the things I would like your comments about is the failure of the scientific community to achieve what DeepMind achieved. So in principle, one of the problems may be that CASP became a metric people were optimizing against. And therefore, they were a little bit limited. Well, it's critical assessment of methods, but in principle, what CASP always compared were the results of the predictions rather than the methods, if you participated. And many people didn't share their method. Actually, the complicated workflow from Yang Zheng you showed is actually one of the more easy ones. Yeah, there were people who were really optimizing against CASP metrics so much that the workflows were 10 times more complex than that one. And these meta methods, and this was really something where I think things were going downhill. Yeah, and I was wondering why all the clever people you didn't mention, Xinbo Xu, who was also in Chicago actually and did a lot of very interesting work. He was the first one to do really a full deep learning workflow for these kind of predictions. But we didn't succeed as a scientific community to do the same thing that a company could do. So why is that? What is the strategic problem we have?
Yeah, thank you, Olaf, for this question, and I fully agree. So this is gonna be probably quite a long answer. So yes, those methods also became way more complicated. And I think a part of the problem was that some people in the CASP community were living just for CASP. So all of their work was dedicated to be better or more creative or whatever in the next CASP. But in my mind, also what it kind of entailed was the assumption that we're never going to solve the problem, right? Like we're chipping away at it, but we're never really going to solve the problem, because otherwise we're going to be in trouble, right? So I think there is something to it. But fair to say, and I'm going to be very open and very transparent about it. So once AlphaFold 1 came out, David Jones was my PhD supervisor, and he was a consultant for DeepMind during the AlphaFold 1 development. So I just called him and I asked him, like, David, like, what happened? Like, how is that possible? And he said, well, it's not that complicated, really. I mean, they have the best engineers in the world, and they have a lot of money. That's exactly what they did. So essentially, David was educating the DeepMind team, but they had, like, we're working, like, we're producing papers with, like, 2 people working on 5 projects, and then you somehow push it through the finish line, and there you go. Sometimes those are great papers, but they had a team of 15 people working for 3 years full time on the problem. And this is exactly what AlphaFold 1 was. There are some articles also that were estimating, like, because they reported, like, how many GPUs they used for AlphaFold 1 training. And I don't want to lie, but I think there was something like, the estimate was it would cost you roughly $10 to $13 million to develop AlphaFold 1, like, including all the person months and all the compute cost and so on. So simply put, DeepMind was extremely well positioned to be able to address this problem. Just from, like, the research freedom perspective, because once they got acquired by Google, and I very purposefully started from alluding to 2014, because DeepMind got acquired by Google in January 2014. It was kind of like a big secret. Like, a lot of people, like, were talking about it, speculating. Then Demis gave his lecture, and one important thing that he mentioned during that lecture, but that I didn't mention, was that they negotiated, like, a full research independence from Google. So Google was entitled to take all of the IP that they created, but they had the absolute freedom to, like, go at whatever they wanted. So there was a team of extremely talented people with limitless money and limitless freedom who decided to solve a problem.
So what is the counterpart then? Well, actually, there were 150 teams, sometimes about 200 teams, who were competing, who probably could accumulate the same amount of, let's say, work time, and they don't have dummies as their scientists as well. So it is just a matter of organizing the community, isn't it?
Well, and that's the beautiful thing, because you know what happened? Actually, the community came together after AlphaFold 2 came out, and they were like, we got to do something about it. And they started doing it, right? There's CoLabFold, there was RosettaFold, like, Rosetta community went at it to try and develop it better. And there are other, like, community-driven solutions to try and address them. I mean, don't get me wrong. I still think that, the best consortium will be kind of less efficient than 15 people working in the same building full time that are paid, 6-figure salaries. But yeah, I mean, the community definitely came together, and how the community works also changed. And I think this is, like, almost equally important as solving this problem, was actually all the different layers of change that it brought, right? So first, like, DeepMind, they were first, but they were not last, right? And Meta and ByteDance and Amazon and Google Research, which is a different company. They're all now in bioinformatics and computational medicine and what have you, right? So really the crossover happened, and that's one very important. But also another thing is happening, right? So researchers and scientists are working with those different companies and different stakeholders of this immense computational power to actually, leverage that and be competitive. And the days of downloading a zip file from an FTP website of some research lab to try and reproduce the program that they created or to use the program that they created, they're long gone, right? And this is also, I think, in large part, well, because times are changing for sure. That's one thing, but also the crossover between academia and industry in those particular areas is happening now much more freely.
Well, do we have any other question or comments from the audience?
I would have one more, actually.
I see Karol Zapała right here, so let me.
I think it should be heard from here. So you said, in some sense, the problem is solved, but I'm curious if it's like really everything solved, or just a good starting point for the new type of the
that's
challenges which I don't know next generation can find to solve.
All right. Yeah. So Karol was asking, is it really solved? Is the problem really solved? Well, no. But I think that the person that's best positioned to actually answer this is sitting right here, Paweł. He's came back from CASP meeting, so like the latest from the community. Maybe Paweł can tell us. But let me give you my take. So I kind of flinched here when it says here protein folding, and I was it's protein structure prediction, right? So that part is not solved, right? The folding part, how proteins fold, we don't know, right? So this is definitely this is not even addressed. Now, in most instances, as long as we have enough evolutionary information, we can reliably predict the protein three-dimensional structure. Now, and even in some of our projects in the group, we're dealing with proteins that there exists just an N of 1 sequences that are related to it. So there is no kind of reference information to orient yourself, how to predict the structure. Those are problematic cases. But now what is also more coming to the forefront are, and this is kind of more biology deep cuts, is, well, proteins are not only kind of single chains, they form different types of machines. So how those proteins interact with one another also impacts their three-dimensional structure of an individual component. AlphaFold deals with this very well in some instances. AlphaFold 3, that I did not mention whatsoever now, is also more kind of developed to start addressing all those problems. But now what happens when protein RNA complexes and all of those different very important, so ribosome, which is absolutely necessary for any cell to live, is essentially a complex of a bunch of RNA and a bunch of proteins. Those are two very different kind of types of biological molecules, and now actually predicting that and those complexes is still very much a big challenge. So there is a lot of open challenges, but I don't know if I want to say as a biologist first, not a biologist first, but in biology there is always definitely answering all of the questions in a given area I don't know if there is a single instance of that where we have kind of like all the questions, all the stones turned, and all the questions answered. So I think, you know, am I expecting another kind of a breakthrough in this space? No, but there are lots and lots and lots of subsequent challenges. And then especially there were some people in the CAST community, and that's also kind of alluding to Olaf, what Olaf was saying, that they were very unhappy when AlphaFold 2 came out. Well, for once, because I think they were like, am I losing my job? But also because they were not happy because this is not the kind of this mechanistic process. Like, do we understand the language of proteins? No, we can predict, given a sequence, we can predict its structure, but we don't know, and like, don't know a lot about like the language of proteins, or like, you know, how to now freely modify those proteins to create whatever you want without those laborious types of simulations and different tests. And second of all, well, we don't understand the intermediates, and then the intermediates or how proteins fold from this extended chain into a nice compact three-dimensional structure. This has significant impacts for a lot of diseases that are related to misfolding of proteins, or, you know, other biological phenomena. So understanding also those pathways, the dynamics of how the protein folds, is very much unopen. And yeah, not yet closed.
Thank you very much. Well, the trouble is that we have reached the time devoted to this seminar. So probably I hope that there will be a chance for discussion offline with Tomasz. So once again, many thanks for this beautiful lecture on with Nobel Prizes in chemistry, which practically are Nobel Prizes in a kind of, let's say, biology, bioinformatics at the same time. So it's also show the evolution in which chemistry is going nowadays. So thank you very much, and I would like to invite you to the third lecture on Nobel Prizes. It will be lecture on Nobel Prize in Physiology and Medicine, and it will be about microRNAs, and it will be given by Dr. Zandil Nar, senior scientist from Concept Life Sciences, from a company. So thank you very much, and welcome. You are welcome to the seminar in a week from now. It will be the last seminar before Christmas, and it will be seminar with number 150 in a week. Thank you, and bye.
Thank you.